Welcome, everyone. Thank you for joining us for Day 2 of Thermo Fisher Asia Pacific Japan Genetic Solutions Tour 2020. My name is Ray, and I'm your moderator for this session. We will be kicking off today's event by continuing various genetic analysis solutions for SARS-CoV-2 research, as well as various emerging applications in this area of research. First of all, we will hear from Dr. Rachel Formosa, who is Senior Director of Global Market Development for the Genetic Sciences Division of Thermo Fisher Scientific. Her focus is on solutions that span real-time PCR, microarray, and sequencing technologies. Rachel has developed expertise in these technologies over her 20 years of experience with genetic analysis technologies, including the past 10 years with applied biosystem products at Thermo Fisher Scientific. For a complete biography on our speaker, please visit the tab at the top of your screen. Today, she will talk about genetic analysis solution for SARS-CoV-2 research, focusing on application in vaccine research and population genomic studies. If any questions arise during the presentation, we encourage you to submit them in the Q&A box. Our speaker will address your questions following her presentation. Now, please join me in welcoming Rachel, and I will turn the presentation over to her.
Thank you for the introduction. Welcome to Day 2 of our Genetic Solutions Tour 2020. I'll kick off our session today about genetic analysis solutions for SARS-CoV-2 research, focusing on applications in vaccine research and host genetics and population genomic studies. As you heard yesterday, Thermo Fisher Scientific has a history of response in public health crises. We've worked hard to make a difference during this current SARS-CoV-2 outbreak, as well as in previous outbreaks of Ebola, H1N1, SARS, and Zika. And while we're talking about our genetic analysis solutions for outbreak research today, Thermo Fisher also provides protective equipment, other scientific research tools like those for protein and cellular analysis, diagnostic tests, and vaccine production and clinical trial logistics. This is fueled by our mission to make the world healthier, cleaner, and safer with our industry-leading scale and unmatched depth of capability. As you know, researchers around the world are diligently working to gain a better understanding of the novel coronavirus with the goal of rapidly identifying future treatment options and possible vaccine targets. We know this research can't wait, and with a comprehensive portfolio of genetic analysis products, Thermo Fisher Scientific is your trusted partner for virus discovery, pathogen detection, epidemiological surveillance, vaccine development, and host genetics to advance your research. To support these efforts, we have a broad range of technologies for genetic analysis, including next-generation sequencing, Sanger sequencing, and fragment analysis by capillary electrophoresis, microarrays, both whole genome genotyping as well as whole transcriptome arrays, and of course, real-time PCR. Since you heard a little bit about our sequencing technologies and the kickoff yesterday, I'm going to provide a bit more background on our microarray and qPCR platforms before talking about specific applications today. So first, our real-time PCR solution. This represents over 25 years of innovation. Here we show our QuantStudio family of real-time PCR instruments, which is our latest innovations in qPCR instrumentation to offer sensitive, specific detection and quantification of nucleic acid targets. We also have a suite of analysis software as well as consumables, qPCR master mixes, and TaqMan assays to power your outbreak research studies. In addition, our microarray portfolio enables rapid epidemiological studies. We have a wide portfolio of instruments, arrays, and free analysis software for both whole genome genotyping and whole transcriptome analysis. Now I want to get into some more detail around application areas. You heard about several key application areas in the kickoff yesterday around virus discovery and sequencing, viral detection, and epidemiological surveillance research. Today I'm going to talk about two areas, vaccine research and host genetics and population genomics studies. With these areas, I'll describe which of our genomic analysis technologies and solutions can help you in your research. First, I'm going to talk about vaccine research. The aim here is to identify protective antigens and study the impact of infection and potential vaccines on immune response. Before we start, we'll review a little bit about our Torrent next generation sequencing portfolio. Our fast, easy-to-use platform offers end-to-end workflows ideal for infectious disease research studies that go beyond single gene analysis. A simple, scalable, and highly automated targeted NGS solutions enable discoveries in viral research, post-response, and vaccine research with the same chemistry and platform, which allows NGS to be more accessible than ever before. It's clear that there's a varied response to SARS-CoV-2 infection, and one question top of mind is what is driving that variability, as well as how that might influence future potential therapies or vaccine response. Human immune systems are complex. One of the drivers of that complexity is BBJ recombination within the immune system that creates millions of different B and T cell receptor sequences, each encoding for antigen specificity. Better understanding of the repertoire of T cell receptors and B cell receptors within a single individual or across a given population will be key to developing new potential therapies. Both innate immunity and adaptive immunity seem to play a role and are important in SARS-CoV-2 research, enabling better understanding of both response to infection as well as to help predict response to future treatments such as vaccines, study potential adverse effects, or quantify immune responses to vaccination. So we've enabled a multidimensional view of T cell and B cell repertoires, delivering accurate TCR and BCR sequencing to assess and quantify immune response for vaccine research. Multiple analyses are enabled using our simple-to-use and highly differentiated informatics suite, from diversity to clone of activation, lineage analysis, and prediction of seroconversion. All areas highly relevant to better understanding SARS-CoV-2. Solutions for both human samples and mouse models are enabled. When studying response to vaccines, it's not just the immune system cells that are potential biomarkers for response to figure therapies. Current research is highlighting aberrant responses in immune response and inflammatory pathways that will be important to understand in relation to vaccine response as well. Ampliseq human transcriptome gene expression analysis offers a fast, affordable workflow to profile over 20,000 RUC transcripts for biomarker identification. Another emerging area of interest is following findings in the field of immuno-oncology that gut microbiome plays an important role in response to targeted therapies or cancer vaccines, and will therefore be critical in understanding response to future SARS-CoV-2 vaccines as well. Recent findings include the role of the gut microbiome in association with disease severity, how dysbiosis can be linked to secondary infection post-SARS-CoV-2, and the antibody response alters with different microbiome profiles. The Ampliseq microbiome health research assay is a targeted approach for accurate, cost-effective species-level profiling. Gene expression microarrays are also used for vaccine research to study host-virus interaction and immune response. Our Clarium V solution allows you to look at the whole transcriptome, while our Clarium S arrays detect exons only. So depending on your depth and throughput needs, we have a microarray solution that covers your vaccine research studies as well. One example of that is using gene expression arrays to look at the expression of ACE2 in human lung tissue across different subpopulations. This research has identified elevated expression of ACE2 in smokers and in those with chronic lung conditions such as COPD, which may help explain why the cases in these subpopulations have experienced more severe symptoms and worse outcomes than other populations. Our other genetic analysis technologies are also used in vaccine research, with real-time PCR being used to verify NGS or microarray gene expression results, and Sanger sequencing and fragment analysis being used for confirmation, as well as cell line authentication and genome editing analysis. Vaccine research is often performed with cultured cells obtained from major repositories of fellow researchers. An estimated 15 to 20% of the time, cells used in experiments have been misidentified or cross-contaminated with another cell line. Although major repositories now authenticate cell line identity, many are calling for all researchers to test and authenticate cell line identity using standard genotyping tests such as STR analysis. This is something that can be done with our solutions, CLA Identifier Plus direct kits on our biosystems genetic analyzers. Finally, for genome editing applications around vaccine research, Sanger sequencing can be used to assess genome editing efficiency. Now I'd like to talk about our second application area, host genetics and population genomic studies, which is the search for potential host contributors to SARS-CoV-2 susceptibility in the hopes of finding genetic markers for COVID-19 progression and biological insights that might point to potential drug targets. When a population is challenged by a pathogen, individual responses can vary greatly depending on their past exposure to infections, environmental and socioeconomic factors, as well as their genetic background. We expect that the genomes we've inherited from our parents will carry common mutations that slightly modify our proteins essential to our defense against infection. So this is what we're aiming to study in host genetics and population genomic studies. Research in host genetics with SARS-CoV-2 infection requires deep insights into genome-wide interactions with the virus. These studies require a whole genome technology to understand the human genetic determinants of COVID-19 susceptibility, severity, and outcomes. Host genetic variation to stratify populations by risk to manage disease, improve outcomes, and contribute to global knowledge of the biology of SARS-CoV-2 infection and disease. And finally, pharmacogenetics to develop research hypotheses for both drug repurposing and identifying druggable targets. So there's really an underlying need for a lower cost, rapid whole genome technology such as microarrays that can offer dense coverage of genes relevant to SARS-CoV-2 infection and medication management. To support this research, we're launching a new Axiom human genotyping array with a SARS-CoV-2 research module that covers various genes and pathways that are implicated in SARS-CoV-2 host genetics. In addition, the array has a large GWAS module, over 820,000 markers, that can serve to conduct association studies to understand the interactions with underlying conditions. And this is something that's available to pre-order now. One example of research to understand human genetics and SARS-CoV-2 infection risk lies within the UK Biobank study, where 500,000 samples were processed in just 18 months on the Applied Biosystems Axiom platform. Some recent insights were recently pulled from this data and published that indicate epidemiological relationships between hypertension, reduced pulmonary function, and pneumonia, which can help identify people at risk for adverse outcomes from SARS-CoV-2 infection. Also, the UK Biobank dataset was used to evaluate the relationship between host genetics and antibody response, uncovering novel genetic determinants of viral antibody response. In addition, human genetics, including genetic analysis for the immune system, plays a critical role in host response to pathogens. As we previously reviewed, when discussing vaccine research, analysis of the human repertoire enables understanding of additional factors that also influence the variants of infection. With end-to-end targeted NGS solutions that include a robust and differentiated informatics suite for analysis of T-cell and B-cell receptor sequencing, populations can be studied to identify biomarkers relevant to SARS-CoV-2 infection progression or potentially to help predict response to future therapies. In fact, Roswell Park Comprehensive Cancer Center has announced that they will be using the portfolio of TCR and BCR assays that I just described to investigate biomarkers to help predict who is likely to develop severe disease and to investigate the potential for a biomarker signature based on T-cell and B-cell receptors in blood samples. This is a critical area of SARS-CoV-2 research where much information remains to be discovered and will have potential impact on the way we move forward in this crisis. So before we wrap up, I wanted to say a few words about our service and support to help keep you running throughout the current coronavirus outbreak. We provide remote support and monitoring options to help you keep your instruments up and running, even when travel may be restricted. So we can help you by including augmented reality and desktop remote support. We have remote monitoring services, and we have cloud-based instrument connectivity so that you can stay connected to your instrument. In addition, we have interactive self-paced education, which is a great way to be learning during this time. Finally, in closing, at Thermo Fisher Scientific, our mission is to enable our customers to make the world healthier, cleaner, and safer. The global COVID-19 outbreak is a powerful reminder of the importance of that mission. We're profoundly aware of our obligation to continue supporting you, our customers, and leverage our capabilities to address this rapidly evolving public health crisis. And we'll be continuing to do so with our solutions. With that, I wanted to thank you for the opportunity to speak today. You can learn more on our website about what I talked about today under thermofisher.com/coronavirus or thermofisher.com/NGS-coronavirus. Thank you.
Thank you, Dr. Formosa, for the outstanding presentation. We will now move into the live Q&A of this presentation. As a reminder, please submit your questions via the Q&A box, and we'll look into it. There are several questions coming through now. How about the first one? Is the B-cell receptor and or T-cell receptor sequencing applicable for mouse? Is mouse spleen cells the right cell type? What is the minimal amount of samples required? Is it DNA or RNA? So it looks like a series of questions regarding the B and T-cell receptor sequencing.
Yes, so we do have mouse assays available for the BCR and TCR sequencing. Actually, we have human and mouse. You can use either or both DNA and RNA, and you can go down to 10 nanograms of nucleic acid input as the minimal amount. Blood is really the common sample type that people are using with us.
Thank you. The second question we have here, is the SARS-CoV-2 array currently available for ordering?
Yes, it is available to order today. You can contact your local sales rep. You can visit the booth or exhibit hall here, get some more information, and you can also go to thermofisher.com/MA coronavirus, and you'll be able to fill out a form there also to get more information about the array, more ordering information about the array.
Thank you. So, Dr. Formosa, I think the next question we have here coming in, what populations are covered on the array? I reckon they are referring to the microarray.
Yes, okay. If this is referring to the Axon SARS-CoV-2 array, this array covers the 5 ancestral populations, so European, Admixed American, African, South Asian, and East Asian populations. It also covers the 26 populations from the 1,000 Genomes Project, and there are markers covering multi-ethnic populations as well.
So let me see our next question here is, you mentioned that there is a potential role for the gut microbiome in the pathogenesis of SARS-CoV-2 virus. Can you elaborate on that?
Yeah, this is still an area of very active research, so there's a lot that still needs to be learned. There have been a number of recent publications studying the correlation of GI disorders with symptom severity, and yet others studying the impact of probiotics in these cases as well. It does seem that there's an association of immune factors known to be elevated during systemic inflammation that correlate both with gut dysbiosis and severity of symptoms, but the mechanisms are not yet well understood. Interestingly, there are other studies showing the impact of specific gut microbiota in response to vaccines, so this is an area of research that will also be important for understanding the potential use of therapeutics or vaccines as well.
Sounds like a research-worthy area to look at. Let me see, the next question we have here is, which of the platform would cater to the investigation of inter-individual differences in response to the viral infections such as SARS-CoV-2? And how can we put it in relation to individual HLA typing?
Yeah, I don't know if we've looked at that specifically yet, not that I'm aware of, but I think that you could use a Sanger sequencing or fragment analysis platform like you do with HLA typing, such as the Seq Studio genetic analyzer, to potentially look at that. But I don't believe it's something that we are investigating currently.
Thank you. The next question we have here is more on vaccine development here. Can we use Oncomine immune repertoire sequencing for developing monoclonal antibodies for SARS-CoV-2?
I'm not sure. I'd have to look into that one.
No worries. So let us address one last question before we transit to the next speaker. What we have here is regarding RNA expression array. Is it designed specific to SARS-CoV-2 infectious pathway, or is it designed to cover the entire transcriptome?
It's designed to cover the whole transcriptome on our MD arrays, not necessarily specific to SARS-CoV-2 infection pathway, but there is some content on there since you're covering the whole transcriptome that would be relevant there.
Thank you very much, Dr. Formosa, for all your sharings on the various avenues to look at SARS-CoV-2 research. Because due to time limitation, we won't be able to address all the questions that's being submitted, but we will look into them and get back to all our audiences about email.
Great. Thank you again for the opportunity to present today.
Thank you very much. With that, I would like to transit to our next speaker, Dr. Soren Alexandersen. Dr. Alexandersen is a veterinary, pathologist, virologist, and epidemiologist all rolled in one. He is specialized in molecular pathogenesis, detection, and control of viral infections since 1982. He has worked around the globe in Denmark, US, England, Canada, and since October 2015, he has moved to Australia, taking up the role as the professor and director of the Geelong Centre for Emerging Infectious Diseases. It is a One Health collaborative centre, including Deakin University, Barwon Health University Hospital at Geelong, and CSIRO Australian Animal Health Laboratory. For a complete biography on our speaker, please visit the tab at the top of your screen. Today, he will share his work with us on the use of Ampliseq panels to study SARS-CoV-2 and the host response to infection. If you have any question arise during the presentation, we encourage you to submit them in the Q&A box. Our speaker will address your questions following his presentation. With that, join me in welcoming Dr. Alexandersen, and I will turn the presentation over to him.
So my name is Soren Alexandersen, and I'm a professor at Deakin University and University Hospital Geelong, and director of the Geelong Center for Emerging Infectious Diseases. I'm going to talk about the use of Ampliseq panels to study the SARS-CoV-2 and the host response to virus infection. The Geelong Center for Emerging Infectious Diseases is located in Geelong in the southeastern part of Australia, as you can see here to the right. So we're in the very most southeastern corner of Australia and about an hour's drive from Melbourne, and a long flight from Winnipeg in Canada, where I worked before. So our center is a One Health center. We work on a healthy environment, healthy animals, and healthy people. We are located at the University Hospital Geelong campus, so we have very close contact to clinicians, and it makes it also easy to get samples rather quickly from, say, patients or any other samples taken at the hospital. As you can see up here, this is our group of people at the center, and I particularly want to acknowledge Anthony Jammings, which is a postdoc working here, and also Taka Bata, which is a PhD student, and they have done most of the work that I'm going to talk about today. So the aim of our center is, through collaboration and coordination, to reduce the impact of emerging infectious diseases on human and animal health in Australia and globally. To do that, we have fairly nice, fairly new laboratories. We started about 4 years ago. This is at PC2, so not high containment, but we can work with clinical samples. And one of our partners is the Australian Animal Health Laboratory, also located here in Geelong, and they have higher containment, so PC3 and PC4, if we need to do any work on pathogens that require higher containment. So a lot of the work that we do at the center is molecular biology, so sequencing, PCR, and so forth. And down here in the right-hand corner, you can see our next generation sequencer, the Iron Torrent machine, and Jesse, another PhD student, is getting ready to load that for some next generation sequencing. So here at GCIT, we used 3 different methods for next generation sequencing. There's the Ampliseq panels that I'm going to talk about today. They, of course, gives targeted sequencing using amplification with multiple PCR amplicons simultaneously. It's fast and it's very sensitive, but you need to have prior sequence information, and of course you need to have a panel designed ahead of time. Currently we use the Ampliseq panels for a human parechovirus, so this is a picornavirus, and we've been working on that for about 4 years now. It's a fairly small virus, so we only need 37 amplicons, and this is a panel that we have designed ourselves and had quite a lot of good success with that panel. We'll have another panel that's for antimicrobial resistance work. There's 815 amplicons in that panel, and it's a panel from Thermo Fisher. And then what I'm going to talk about today in particular is the SARS-CoV-2 panel, which has 237 amplicons targeting the virus and another 5 amplicons targeting human genes as controls. We're also working with Thermo Fisher on an inflammation panel that has 683 amplicons, and we are planning on using their immune response and the T-cell and B-cell receptor panels as well. So that's in the pipeline. In addition to the Ampliseq panels, we also do other types of next-generation sequencing, including total DNA extraction and sharing. That, of course, gives an unbiased and more or less quantitative reads, but it's not very sensitive. We currently use that for antimicrobial resistance, for microbiome work, bacterial isolates, and so forth. And the third method that we're using and have had quite a bit of success with as well and can complement the Ampliseq panels is virus or particle enrichment and unspecific amplification of all protected DNA and RNA, in which case we can look pretty much for any pathogen, in particular viruses, but other microbes can be looked at as well, as the ribosomes from various microbes are also concentrated and purified by this method. I should stress that all methods require specific analysis of the sequence reads, and one thing with the Ampliseq panel is that the analysis is usually easier than for the other methods. As I said, we have been working on another virus for at least 4 years now. It's a paragovirus, or a small picornavirus that affects very young babies, and more or less came to Australia back in 2012. We need very accurate sequencing for these viruses because we're basically following them by single nucleotide changes, as you can see here, and we have had pretty much outbreaks since 2012, pretty much every 2 years. And as I said, we have our own panels, Ampliseq panels, for looking at this particular virus, and we need very accurate sequencing as we follow the virus as it evolves with single nucleotide changes. Coronaviruses, and of course today we're going to talk about the SARS-CoV-2 and the alpha and the beta coronaviruses. So those are the viruses that traditionally comes from bats. But there are other coronaviruses that we have been following for some time. A lot of people look at bats and host switching of bat viruses and other mammalian viruses, but we have been looking quite a bit on birds because they also have coronaviruses. There's the traditional gamma coronaviruses, which appear only to affect birds, but not too many years ago, another genus of coronaviruses were discovered in birds called delta coronaviruses, and it turns out that some of those delta coronaviruses can also jump species, so jump from birds, and they jumped into small carnivores in Asia, and more recently into pigs, and they made it into North America back in 2013, 2014. So there's some evidence that also some of these coronaviruses, at least the Delta coronaviruses, may also be able to jump into mammals and perhaps eventually into humans. But today we're going to talk about beta coronavirus, so most likely with an origin in bats, and we're specifically going to talk about the SARS-CoV-2. As I said, we have been looking a bit on these bird viruses, the Delta coronaviruses, and they are present in Australia, and we can follow them, and they seem to be both in resident birds, such as ducks, but also in migrating birds, and we can see that they actually interchange over long distances, and they migrate over long distances, and we can see that we have viruses here in the southeastern part of Australia that are very, very similar to viruses we find in migrating birds up in Siberia, Alaska, and down the west coast and the east coast of the US. So again, birds can also migrate over long distances, and they can kind of transmit these Delta coronaviruses among them. And as I said, there is some evidence that at least in rare cases, some of those Delta coronaviruses can also jump into mammals. But today we're going to talk about beta coronavirus and SARS-CoV-2, specifically using the Thermo Fisher Iron Ampliseq SARS-CoV-2 panel. So in terms of samples, so we have the panel and we have samples. So the samples we have been working on are basically leftover oropharyngeal swab material. So they're swabs taken from individuals, and they are traditional kind of diagnostic samples. They're taken here in the greater Geelong area, and they're tested at the Bowen Health Australian Rickettsial Reference Laboratory or the Australian Clinical Labs so that we know whether they're positives or negatives. But as I said, there are routine samples, and they've been used for initial testing, and they're also needed for future studies. So we have very small sample volumes and sometimes not optimal quality as they've been tested several times and maybe frozen several times as well. So as we have very little volume, we also have to extract nucleic acids from very small volumes. And as we need them for several different studies, maybe elute them in a higher volume that you would normally do. So we have very low concentration of nucleic acids and very limited sample volumes. So far, we have collected 55 samples, including 20 samples that are positive or in the dubious range. So these are from kind of routine diagnostic testing, so many of them are very weak positive. So we have attempted this far the Ampliseq sequencing with the panel of a total of 15 samples, so 14 positive and a negative control. Of the 15 samples, 2 were not taken further as they did not amplify with the Ampliseq panel, and there were 2 very poor samples, so just barely positive by PCR. So overall, we have taken 12 positive samples and 1 negative control sample through NGS and created a total of 58 million reads on 530 chips. And I should say before I go to the positive samples that the negative sample is negative as expected. Of the 12 positive or weak positive samples, dubious samples, we have got near full length, good quality sequence from 6 of them, and they have been submitted to GISAID, so that's the global influenza and now also coronavirus database, and partial sporadic sequence of the other 6. The 6 samples that only provided partial or sporadic sequence either had a very high CT, so a very low virus load, or for 2 samples which actually had a low CT, so a high virus load, and gave poor sequence. We now know that they're poor sample handling and storage, so that we have degraded RNA in those samples. So they still give a good low CT in the PCR, but the sequencing gives short reads, and we only get kind of sporadic sequence coverage. So overall, it works very well with good quality samples with a CT in our hands at or below 31. You can probably go above 31 if you have good quality samples, but as I said, we have only access to kind of well-used diagnostic samples, and they can't be degraded. So we have submitted this work. We have submitted it to a journal, and it's available in mid-archive. You have the title there, Sequence Analysis of Travel-Related SARS-CoV-2 Cases in the Greater Geelong region, Australia. And as I said, that is freely available on mid-archive. To give a bit more detail there, of the 6 good quality sequences that we assembled from these samples, 4 of them were essentially full length, and they had a CT value, so a PCR load of 18 to 31. So again, it's about having good quality samples, maybe more than having a lot of virus in there. One sequence was missing 46 nucleotides, so a small gap in there, as you have several amplicons. If you have a bit of degradation of your sample, maybe you might lose a little bit of the sequence, but as I said, for 4 of them, 4 out of 6, basically the complete sequence, 1 missing a little bit, and for 1, we had about 1.76% of the genome reported as ends. And if you look at GISAID, you will see that that's not unusual, that you might miss a few bases along the genome. As I said, the good quality samples, the panel provided length sequence, and basically only missing 42 nucleotides at the 5' end, and 28 nucleotides and the poly-A tail in the 3' end. So this is where the kind of the terminal primers are located. And although you might get the sequence as such, it's part of the sequence, it's part of the primers, and really you shouldn't really trust them. And this is more or less a general phenomenon, and you would see in places like GISAID that they usually mask or remove the ends of the sequences anyway, since they're usually not reliable. Assembly of our sequences was initially done using the plugins, so the Torrent, Iron Torrent plugins, Perma, and COVID-19 Annotate SNP plugins. So those are the ones that can give you the differences of your sequence as compared to the reference, and also assemble the near full length genome. This is easy, it works very well. However, it only resembles good sequence for high quality, high coverage samples. And in my opinion, or our opinion, you should manually check it, and you should trim the ends and so forth. So it does assemble near full length sequence of high quality, but again, I think it is a good idea to check it manually afterwards. So when we assemble these sequences, we often use other free programs. So we use free programs such as IGV, the Integrated Genomic Viewer, and also MEGA, also another free program. IGV is particularly suitable for this, as you can kind of get a visual view of the quality of your sequence. And if you look here, the top sequence here, you see that there's very variable coverage across the genome, and the reads are very, very short. So this is a poor sequence on a poor sample, and we know the reason. Now we know the reason why it gave poor sequence, which was basically because that sample had been put in water rather than in transport medium. This is the second one here. It's a good sequence, very good coverage, basically from one end of the genome to the other one, good coverage, and we see that there's a few sequence differences as compared to the reference. So from that one, you can assemble pretty much a full virus genome. The two lower ones are also fairly good quality, but again, maybe not able to assemble the full genome, but very, very close to the full genome. Then when you have assembled the genome, of course, you can use it for different types of comparison, either using NCBI or various programs or downloading available sequences. And that's both NextStrain, that's GIS8. And for SARS-CoV-2, there's, last time I looked, about 50,000 near full-length genomes available. So there's a lot of phylogeny and comparison you can do. This is just showing a very simple comparison, as we were interested in seeing whether the travel history of our individuals, our positive tested people, they have been traveling, so they were returned traveler. We knew where they had returned from. And using this type of analysis, we can then look at whether that fits the sequence of the virus fit with where people have been traveling back from. And this particular individual that you see here had returned from the United Kingdom, and the closest sequences are also the sequences that have been deposited in the database from the United Kingdom. So this is just to show a few examples. Another one, we know that it was a returned traveler from Germany and Europe, Central Europe in general. And again, the virus is closely related to viruses from Germany and Netherlands. So again, using the sequence, the very accurate sequence you get, you can start to look at these type of things. Just the final one. So many things you can do when you have assembled the virus sequences. But of course, you have to have accurate sequence to look at these type of analyses. So just towards the end here, I also want to talk a little bit about other panels. So we're talking about here the virus panels for the SARS-CoV-2 and assemble of full-length virus sequences from the same samples. And as I said, we have very low volumes, so very little nucleic acids that we can extract from these samples. But again, using panels from Thermo Fisher, Ampliseq, you can look at other things. And here we are looking at the host response to the infection. So we have a number of positive samples, SARS-CoV-2 positive samples, and a number of SARS-CoV-2 negative samples, and these are swab samples. It's the same nucleic acids that we use to assemble the full virus. And we can look using the so-called Ampliseq inflammation panel as to the host response. So here we have about 683 different amplicons or different genes targeted by this inflammation panel, and I'm not going to go into details, but it's basically doing the same. It's amplifying messenger RNA for these various inflammatory genes or genes associated with inflammation, and we can then get reads, next generation sequencing reads, that we can then look at the abundance of those reads for the various genes in the various samples and compare them. And this is just showing a plot at the top. And these are when we look at, as I said, 683 genes, there's about 250 or 253 genes in these particular swab samples that we can detect by this method. And these are plotted here on the x-axis, and the levels of them in the individual samples up at the y-axis. And you can see that certain samples have certain genes that kind of sticks out. What is shown at the bottom here is around 0 here is where positive samples, so SARS-CoV-2 positive samples and SARS-CoV-2 negative samples have the same level of these particular genes. And again, you can see for SARS-CoV-2, some genes are upregulated and a number of genes are downregulated in the SARS-CoV-2 infected individuals. This is something that we started very recently, but it shows that again on the small sample volumes and even on these type of swabs, you can start to look at the host immune response or inflammatory response to the infection. There are additional panels that we are looking into, such as the Thermo Fisher immune response panel, and also the B-cell receptor and the T-cell receptor panels, so that we can fully characterize the virus infection, but also the host response to the infection. So at the very end here, this might seem complicated, but when we do the panel, so when we do the virus panel looking for the full virus genome, as it turns out, the panel is designed to give the full SARS-CoV-2 virus sequence, so the genomic sequence of the virus. As it turns out, as you can see here at the left, coronaviruses do have what's called a leader. So for the SARS-CoV-2, this is a sequence of about 70 nucleotides at the very 5' end of the virus. So the panel is designed so that it has primers in the leader, and then of course it has primers that sits in the open reading frame 1, close to those primers there that are supposed to give sequence from the virus genome itself. As it turns out, the way the virus replicates in the cell, it makes what's called subgenomic RNAs. And those subgenomic RNAs, which you can see here on the right, of which there's 10, supposed to be 10, we can only find 8 in vivo, but the virus can probably make at least 10 of these subgenomic RNAs. All the subgenomic RNAs have a common leader. So they actually have this 5' leader is connected to the various open reading frames. So in order for the virus to make the spike protein, for instance, or the various other proteins down here in the 3' third of the virus, it needs to make these subgenomic RNAs. Each of the subgenomic RNA, as I said, has exactly the same sequence. So it's basically this piece of the sequence sitting in front of the gene. So that means that when you do Ampliseq sequencing, or if you do amplicon sequencing with primers that have sequences in the leader, those primers will actually also sit in what you see here as a black box in front of each of the subgenomic RNAs. These subgenomic RNAs was initially thought to be more or less unstable, but it turns out that they're fairly stable and found mainly when you find the virus, you will also find the subgenomic RNAs. And with this particular panel, and as I said, with any amplicon-based strategy for which you have primers in the leader, and if you then also have downstream primers that sits in these individual genes, which you will have because the amplicon-based methods basically cover the whole genome. So you will have a reverse primer sitting pretty much in any part of the genome, and you're going to have these early primers forward. They would sit in front of each of these subgenomic RNAs. So as it turns out, and what we found out fairly recently, is that when you use the amplicon sequencing, you do not only get the virus genome, but you can actually also map the individual subgenomic RNAs. This requires slightly different analysis of the reads, because when you normally assemble it, you assemble it against a reference, and you're going to assume that it's the virus, the full virus genome. But because these are kind of put together by a specific open reading frame and a leader sequence, it just requires a slightly different analysis of the sequence reads, but you're able to actually find the various subgenomic RNAs and the abundance of those RNAs in exactly the same panel run. So it's basically using the same panel, it's using the same data, will give the full virus sequence, but it will also give the presence and abundance of these subgenomic RNAs. So I only have left to give the acknowledgement, in particular to Anthony and Targa, for doing the work, to other people in the lab, to the clinicians and the diagnostic labs, to Thermo Fisher, and to the various people that have funded our work. Thank you.
Thank you. Thank you, Dr. Alexandersen, for the outstanding presentation. We will now move into the live Q&A portion of the presentation. As a reminder, please submit your questions by the Q&A box, and we will look into them. There are a number of questions coming through right now. Our first question is, why is there a need to use a panel to conduct the study instead of a whole genome sequence?
So, well, as I explained, with our samples at least, we have very little sample material, and in many of them, we have a very low virus load. So in order to get enough or to get good sequence covering the whole virus, you would usually have to amplify.
So, Dr. Alexandersen, I noticed that one of the methods that was used was ultracentrifugation. Is there any downsides of using that?
Well, one of the methods we use, that's not for the panel, that's for the unbiased sequencing and concentration of virus particles or particles. We use ultracentrifugation. For the panels or the Ampliseq panels or panel amplification, we do not use ultracentrifugation.
Thank you. Going to the next question. How important are these epidemiology studies in driving lockdown decision in the country? Will lab be processing more of these samples as a requirement for the national program to actually look into making any decision?
Well, for the national program, it's entirely based, at least to my knowledge, it's entirely based on PCR testing. So diagnostic PCR testing, and then decisions made from that. So, the studies that we do in terms of the specific virus sequence or, say, the inflammatory response has not have any impact on the actual testing and response.
Okay, the next question. Is it known that other coronavirus present in animals apart from bats that can affect humans?
Well, it's not known that there are any, but I mean, several of them, the ones we already have, they did not all come from bats, or maybe they originally came from bats, but maybe they went through rodents or, say, bovines or cattle. So clearly, these viruses can change host, so they may be able to get into another mammal, maybe initially, and then eventually into humans. So it may not happen just kind of right away, and they may not be present in a bat at this moment, but there are clearly viruses in bats and perhaps also in birds that eventually, given the right opportunity and maybe bad luck or good luck from the virus point of view, may make it into another mammal or into a mammal if it's from a bird, and then eventually it may end up into humans.
Thank you. So the next question we have here is more on public health awareness. One Health has long been a global advocacy in terms of viral surveillance and interrelationship between animals, human, and environment. From your point of view, how can we help to strengthen this public awareness?
Well, I think, being a veterinarian and having worked with animals for many, many years and in the One Health space, One Health and kind of things jumping from animals to people is not new at all. And as a veterinarian, is something we've been concerned about for years and years and years. I mean, basically started with bovine tuberculosis and brucellosis more than 100 years ago. So I think from the veterinary profession or the veterinary point of view, it's a no-brainer in a sense. But I think to convince the human health side or the public health side because it seems to be forgotten every time. It seems to be remembered every time things happen, but then in the meantime, in the interim or between episodes, it seems to be forgotten. And in reality, humans are just another species. Everything else, I guess you call it animals, but it's kind of it's a continuum of all kind of species, including humans, and clearly we are not that different.
Probably continuing from the One Health conversation, do you reckon that because One Health is more of a global initiative in a regional level such as Asia Pacific, because we have different dynamics, is there a need to have some kind of organization or consortium that actually advocates the same message from One Health?
I think it would definitely be an advantage to have some organization that can, well, can give the message, but it's also about having people actually work closely together, so that maybe, let's say I represent more the lab side or the laboratory side of things, that very often you could have a better integration between human public health and animal health, because the methods and the knowledge are often very, very similar. And quite often what we know from the animals field, both in terms of diagnostics, but also in terms of control and in terms of maybe action, can be, to a certain extent, be kind of extended into when we have a public health emergency. So I think there is a lot to learn, or at least a lot to transfer or to interact between the animal health side and the human health side, both in terms of lab diagnostics, but also in terms of knowledge of infections, because many times we think that an infection in people are very, very special, but very often there are very similar infections in animals that we already know of. And if we talk coronaviruses, we've known about coronaviruses, well, in people, but we've known about them in animals, including livestock, for many, many years.
So it sounds like such an organization does not exist here, but does it exist in a national level in Australia, Australasia?
Does it exist? I mean, well, we have the Geelong Center, but that's a fairly small center. And I think most people do talk about One Health, but to actually get it to work is very difficult, because there are clearly silos, human health silos, and maybe also animal health silos. I think from the animal health side and from the veterinary side, we're very prepared to work with the human health side. But as I said, and maybe that's just my personal view, it's often more difficult to get the human health side to actually accept or maybe come to the animal side, at least in peacetime. And by peacetime, I mean when we don't have an outbreak.
Good point. The next question I have here is more on the viral load. Does looking at the subgenomic region in SARS-CoV-2 correlate with viral load?
Well, indirectly, the more viral load, you also seem to have more subgenomic RNAs. So it's not. I think that the message here is more that they're there. The subgenomic RNAs are there, which may be slightly a surprise, but they seem to be there even late in infection. And from at least for our samples, when we can detect the virus or the virus RNA, excuse me, we can also detect the subgenomic RNA. So there is indirectly a correlation. The more virus, at least in our samples, you also have more of the subgenomics. But even when you have fairly low level of virus, you can still detect them at a low level, these subgenomic RNAs.
Probably the last question I have here is, you spent a bit of time on the lead sequence that was on the last slide. What do you think are the impact of this lead sequence in terms of pathogenesis or even viral load? Because it seems a very interesting finding.
Yeah, so the leader sequence, all the coronaviruses have a leader sequence around the 70 nucleotides long. It can vary a little bit, maybe from 50 to 100 nucleotides. And it's not only the coronaviruses, also other viruses have the same. And I guess they need to have it in order to make these subgenomic RNAs, and they evolved kind of into having a very, very nice way of making their subgenomic RNAs. And if they couldn't, so if they didn't make those, they wouldn't make the proteins from the whole 3' end of the virus. So it means that it will not make any spike protein, which is, of course is the receptor binding protein, and also the nucleoprotein, which is a very important protein for the coronaviruses. So yes, they have the leader, and it sits there, and I guess in the genomic RNA, it may look as if it's not really doing anything, but it is actually very important because it has to sit there in the front of these subgenomic RNAs. And it's basically a way that the virus, at least indirectly, makes its messenger RNAs, these subgenomic RNAs, and also the whole virus RNA are basically looking like messenger RNAs. So it may kind of partly fool the cell to think it's a normal cellular messenger RNA, because, well, I don't have time to get into that, but it's not just the leader, but it's the way it makes these subgenomic RNAs, and it's also kind of the way that it gets proteins to sit on it. So it basically, when it's replicating and making them, it's hiding it from the cell, and when it gets into the cytoplasm in order to make proteins, it's more or less looks like a normal cellular messenger RNA. So the cell, in a sense, is getting tricked into not responding with a severe immune response.
Thank you, Dr. Alexandersen. I think that's all the time we have for now. However, thank you, and as a final reminder to all our audience, any questions that were submitted and not answered today, we will get back to all of you via email. And I would like to thank Dr. Alexandersen for his time, for sharing with us on his insights on the SARS-CoV-2 and the epidemiology of that. So thank you for all the questions and all the speakers for spending their time with us. We will now take a 15-minute break before we start the next session. Check out various application tracks that may be of your interest at our booth. Please go and have coffee, visit the booth, learn more about exciting promotions that are available to you, where you can ask questions to our Thermo Fisher representative at each of the segment booths. Please also participate in the scavenger hunting game. Look for icons in the environment to win prizes. There will be a pop-up survey appearing in a minute. Please help us to complete the survey. And thank you, and look forward to speak to you again 15 minutes later. Thank you very much.